A large portion of the agricultural production in the United States is within close proximity to St. Louis. The city has begun to attract national attention as a leading center of the biotechnology industry.
This is Science Friday from NPR. I'm Ira Flata. We're talking this hour about biotech crops. Richard Sayre is director of the Cassava Plus program at the Danforth Plant Science Center here in St. Louis. Happy to be here.
When the St. Louis Science Center hosted a live national broadcast of NPR's Science Friday, one of the topics was the Cassava Plus team at the Danforth Plant Science Center.
So most of us know cassava probably as tapioca. It's also known as yucca. It's a crop that originated in Brazil, moved to Africa, where it's a very important crop. It grows in very poor soils, and it can be relied upon to provide food for a long period of time. Most importantly, you can leave the roots in the soil for up to 3 years, come back and harvest them piecemeal as you need food. And so it extends the growing season effectively over 3 years.
In 2008, I visited the center and met some of the other team members working to improve the nutritional value of this important food staple. For the past 10 years, the Danforth Plant Science Center has been conducting research on how plants can alleviate world hunger, fight disease, and improve the quality of human life.
We are long-term optimists about the future of mankind, about the future of the earth, the globe, knowing that there are challenges, but we also are confident that the intellectual pursuits chemists are involved in, that physicists are involved in, will all come together to ensure that we do things right in the future.
One important project focuses on a tropical plant called the cassava, the major food crop of Africa. Every day, 800 million people on Earth rely upon the root of the cassava for sustenance. It grows all year, is simple to cultivate, drought-resistant, and can be prepared in a number of ways for meals. But it has some serious drawbacks, which scientists here are working to eliminate.
Cassava's roots are very full of starch, meaning energy, but they don't have proteins, they don't have vitamin A in particular, and they don't have what we call zinc and iron. This is what we call micronutrient. All these elements are extremely important for human health.
Another problem is its susceptibility to disease.
These leaves here are virus-free, and they look like a virus-free plant. And this is what we would like to see all cassava looking like in Africa, looking like plants like this. And on the opposite, you can look at this particular plant here, and the leaves are small, they are very narrow, they are yellowish, what we call the nodes are short, etc. And this plant will be dwarfed compared to a plant like this. So the role of this study is really to investigate in detail, in molecular detail, the makeup of the population of these viruses in order to understand and investigate the possibility to block them.
The process of developing a new and improved cassava plant has taken years, evolving with new discoveries in the field of biogenetic technology and creation of a process in which DNA with desired characteristics is integrated into the genome of a plant intended for improvement.
We start with cuttings from a plant, very much the way you would do cuttings in your own home to make several plants, and we can produce plants from those cuttings. Then we take leaf lobes, which are unexpanded tiny leaves, and from these we produce a certain kind of cell which is capable of producing a whole new plant. These cells then, through a complex process, we can get genes into these cells, genes that we're interested in doing research on in our laboratory, and from these new cells we can produce a whole new plant. This plant is now an improved plant with the traits that we have put in, and now we end up with a plant very much like we started with. However, now this one has an improved trait, and this is what we're doing in the lab.
We teach the plant, we inoculate the plant, so then when the virus is coming, this sequence of DNA is already known by the plant, and the plant has more time, is prepared to control these viruses better. And if you do this job very well, then the winner will be the plant.
The next step is to grow the plants they hope are improved in the greenhouses, where they are injected with viruses to see if they get sick. The success rate for making the cassava more nutritious is high, But inoculating plants against viruses is a much more difficult and gradual process because viruses are so complex.
The question is this particular planted virus resistant? Unfortunately, this technology is not 100% efficient. If we make about 1,600 different plants per year, and last year in 2007 we have selected less than 25 plants that are highly resistant to the viruses that we are checking for.
So it's a long road from scientific discovery in a laboratory to practical applications of those discoveries. It could be another 7 years of research and experimentation before Africa benefits widely from this more nutritious, virus-resistant source of food.
So we are not done yet. We still have to, improve our technology. We still have to transfer these technologies to Africa. I'm very happy because we have made, it has been a long road, very slow process, but now we see things speeding up.
Meanwhile, the Danforth Plant Science Center continues to work on other projects, exploring plants' potential to resist pests, remove pollutants from soil, prevent human sickness, provide biofuels, and offer raw materials for industry. Goals consistent with the center's mission to improve the human condition.